Hey team, what a day it's been. It's 2:42 and I've already went paddleboarding with some dolphins. That was nice. And then I caught a flounder, a large flounder. So I caught my dinner. Yep. Well, we're going to continue on. I'm going to share my screen with you. This is the second video. Eventually I'll lose track and I won't be able to say this is the whatever number of videos. So basically you're just watching these videos in order. You can always go back and re-watch them. Today we're going to finish up plant tissues and probably move into organs, but we'll see how it goes. This is going to be about approximately 1 hour video. So we'll cover a good bit of information. Okay. And sorry, I'm moving things around a lot here now all of a sudden. Let's see. Nope, that's not it. Here we go. There we go. Sorry about that. I just wanted to show you where this PowerPoint that we're working on is. This is module 2, which says plants, and we're still in the plant tissues PowerPoint. So that's where we are at. And just, you know, kind of quickly, we did simple tissues last time, talked about what their function was and what they looked like. There's some parenchyma with very thin cell walls, storage of food and water. Collenchyma, flexible support. You can see the irregular thickenings of the cell walls and the collagenous or the collagen fibers that are present that gives them flexibility. And then these bundles here are sclerenchyma, these sort of rebars, very thick lignified cell walls. And we talked about sclerenchyma. Sclerids are stone cells, and we talked extensively about fibers. And then we got into complex tissues. We talked about xylem, and we talked about phloem. And so remember, xylem moves the water up. There's no, it's not a two-way street. Phloem, and there are two different kinds of xylem, tracheids, and vessels. And then phloem is, in fact, a two-way street. The sugar can move up or down through the sieve cells. And then remember, these are living cells, but they're enucleate, meaning they don't have a nucleus. Each of the sieve cells has a cell associated with it that does have a nucleus that we call the companion cell. So just to remember, xylem is not living at maturity, but phloem is. Okay. Talk a little bit about secretory tissue. Plants make all kinds of secretory substances that are, and by secretory, what we mean is it's a substance that's secreted to the outside of the cell. And you could compare that with animals. Animals have secretory tissue. Think about mammary glands in mammals. They produce milk and they secrete it out of the cells. Well, plants produce a whole host of different secretory compounds. What you're seeing here is a rather notorious one because this is Papaver somniferum, or the opium poppy. And you can see this latex, this white milky latex that's oozing out. This latex ends up being about 30% morphine. And so it's a potent pain killer. Okay, we'll talk a little more about that in a second. Let's make a list of different kinds of secretory substances that plants might make. Latex is important, and it's important for a variety of reasons. We use it, it's useful. I don't know why I put a T there. Didn't do very well, sorry. Let's try this. T-E-X. Maybe that's how I had it spelled before, but it looked funny. Examples: rubber, right? So we use rubber to make tires and other things, right? Latex gloves, for example. Condoms are made from these secretory substances of plants. And then when you start thinking about Papaver somniferum, this is the opium poppy. And so in this case, the latex is used medicinally or in some cases recreationally. So a little bit of information about opioids. One thing that's fascinating is that your brain has receptors that perfectly fit the opioid molecules, and so these molecules fit in these receptors, and this is what accounts for the pain-killing effect, and also the other effects of the psychoactive effects of the opioid. Somniferum means to rest or restful. So for a long time, scientists wondered, why in the world does your brain have some receptor that this plant molecule fits so well in? And it was discovered that we actually, it's because we make our own morphine. We make this stuff called endorphin. So how does opium work, or how do opioids work? And what are some opioids? Well, morphine, heroin, codeine, these are all opioids. And then there are lots of synthetic ones like Oxycontin, hydrocodone, et cetera. So endorphin is literally endogenous morphine. And the thing about it is your body is constantly making a little bit of it at all times. If endorphin is kind of known as the the feel-good, one of the feel-good neurotransmitters. So when you unfortunately use opioids, there's this feedback mechanism, and when you take synthetic opioids or even natural opioids like opium, your brain immediately thinks it's producing too much endorphin. And in response, it shuts its own natural endorphin production down. This is why opioids are so incredibly addictive. You cannot live without this molecule. But if you abuse opioids, your body stops making this molecule, and at that point, you have some hardcore addiction on your hands. And then, of course, if you stop taking the synthetic version of the neurotransmitter, you're going to go through terrible withdrawal situations. So, the take-home message there is, although opioids have their place as painkillers, they can be very dangerous because they are such addictive and potential for abuse drugs. Okay. It is illegal to grow opium poppies, although they're commonly found in, they can be found just in wildflower mixes that you might sprinkle in your yard. You can also just go to the food co-op and buy poppy seeds, and those end up being most likely opium poppies because they're growing the opium. They're growing the poppies for opium, and then they're also harvesting the seeds. Okay, so what is opium? It's just that latex that you saw oozing out of the plant. And so the way that they harvest it is they make these little lacerations in the fruit. This is the fruit of the poppy. And then they'll come through and scrape this latex off 2 or 3 times a day. And that would be raw opium. And then from there, you can refine it into heroin or morphine. Okay? So there's that. What are some other secretory substances? Oils, resins. These are fat-soluble, sticky things. Some good examples. Well, as far as oils go, humans become obsessed with these oils especially the aromatic ones. I mean, obviously we use some oils to cook, but think about aromatic oils like peppermint. Another one is THC, tetrahydrocannabinol. It's a resin. And, you know, when we start looking at these oils and resins that are produced by plants, we immediately would wonder why. Why are the plants producing these chemicals? They're large compounds, they're expensive to make. Most likely anti-herbivory. What does that mean? Herbivore, right? Eats plants. So these compounds tend to be distasteful to herbivores, to things that might come and eat them, like insects or even mammals like deer, right? So, you know, peppermint oil is a good example. Also, the glands around the female flowers of cannabis produce THC, right? And it's believed that this sticky resin is there to prevent herbivory of the seeds ultimately. Okay. Now humans are obsessed with these. Go to Earth Fair and look at all the essential oils. There's probably 75 different ones, right? Where we extract these oils and we use them because they smell good to us. Of course, THC, you know it's a psychoactive chemical, so it's used as a drug. It's the active ingredient in cannabis. Okay. There's also CBD, which is another type of resin that's produced, or oil. So that gives you kind of an idea. You know, these latex and oils and resins, they can be used in industry, they can be used you know, to make tea or peppermint tea, or they can be used as recreational drugs in the case of THC, or medicinal drugs, as in the case of THC and opium latex. Okay. So that's pretty good for secretory substances for plants. Okay. Moving right along. Now we want to talk about meristematic tissue. And this is kind of a funny word. There are lots of funny words in botany, and I don't know, for some reason the word meristem seems kind of silly. What is meristematic tissue? Meristematic tissues are regions of actively dividing cells in plants. And so you start thinking about how plants grow. Plants grow from the tips of the plant, not from where the plant comes up out of the ground, right? And so what you're looking at here is the tip of a coleus plant. And you can see this is a leaf, and these are very small leaves here, and this is another leaf. And these mounds that you see here are the buds, and inside of the bud is what's called an apical meristem. And this is a region of actively dividing cells. Remember what that process is, right? How do we make more cells asexually? What's the process? Mitosis, right? So this is a region of mitosis. And then here are some lateral buds a little lower down, which are also meristems. So we have different kinds of meristems. The apical meristem increases length of the stem. So as these cells do mitosis and divide, the stem grows in this direction, okay? So that's an example. Same thing in the root. In the root, we have, and when we talk about organs, we'll talk more about roots. This is the root apical meristem. Same thing, it increases the length of the root. Okay, so we have increase in length. SAMs and RAMs. SAM is shoot apical meristem and RAM is root apical meristem. But then it gets pretty complicated in a woody stem, right? because we have, didn't mean to hit that button. There we go. Because we have these different regions of tissue. And there are tissues that form a cylinder. And this is the one, what have I done? I've lost my ability to write. That's unfortunate. I don't know why that is. Let's see what's happened here. Thought I took a screenshot of it, but maybe not. There we go. Okay. So the important word in this image for now, and we're going to come back to this, but the important word is vascular cambium. Okay. Anytime you see the word cambium, the cambium is a meristem, but it increases girth. So it's laying cells down in this direction, okay? And in doing so, it makes the stem bigger. And in this case, we're looking at what we would presume would be like the trunk of a tree, right? And so here's the thing, the vascular cambium makes more vascular tissue. And if you recall, the vascular tissue is the xylem and the phloem. And so here's the xylem, in here, the wood, and then it turns out that the phloem is right in this region. And I'm going to actually draw this so that we can see a little bit better what's going on. Let's do that. Okay, so let's say we have a tree, and as you may know, you can count the rings of xylem and know kind of how old the tree is, right? And we have this region right here that I'll do in a different color. And I'm going to label all this stuff here in a second, don't worry. And then we have this region right here. It's a little hard to draw. So you get the idea. And then we have all these different colors. It's kind of crazy because we have all these different layers. And we're going to revisit this when we do organs. This is obviously the cross section through a stem, a woody stem. And then we'll go back to black, and here's the bark. Okay. So no time like the present to learn the different layers of tissue here, because we're talking about these cambial layers. And I'm going to label those. This is the vascular cambium. And each year during the growing season, whenever that is, it makes more xylem and phloem, which is the vascular tissue, right? Okay, and there's a rule about how it lays this xylem and phloem down, and the rule is it always makes the xylem to the inside and the phloem to the outside. And so what that means is each year of growth, there'll be another layer of xylem laid down to the inside of this vascular cambium and another layer of phloem laid down to the outside. So xylem inside and phloem outside. But we have another cambial layer here, and it's this one right here. And this one is called the cork cambium. And the cork cambium makes more cork each year. What's cork? Well, you think of it as the bark, right? So this is the cork right here. Okay, so digest all that for a second. Think about these different layers. What is all this tissue? Well, in black on the inside, this is the xylem. And then blue must be the phloem because it's on the outside of the vascular cambium. And then the cork cambium, it makes the cork. Anytime you see this word cambium, anytime you see this word, think it's a meristem that increases girth. And now think about this. This is a cross section, but The vascular cambium literally forms a cylinder that goes all the way up the trunk of a tree, right? Goes into every branch and every stem and even into the leaves. And these are regions that can do mitosis and make new cells. Okay. Lots of cool stuff here. And now we can look at this image again and see. We'll talk when we do stems, and we'll talk about heartwood and sapwood and all that. Okay. All right, y'all. That's good. We're moving along, and just note that that is the last slide in this particular PowerPoint. So what we're going to do now is we're going to move into the next PowerPoint, which is a plant organs PowerPoint. So let's look at that. And you can see there is some required reading for you. You can read plant cells, and if you haven't already, read plant tissues. And then here's the plant organs PowerPoint. There's some required reading about plant organs. Now, when you start thinking about plants, you realize, well, plants aren't as complicated as animals. Think of all the organs in an animal, right? I mean, you got your heart and your lungs and your liver and your skin, right? Plants are pretty straightforward. We have roots. We can break plants up into vegetative and reproductive organs. And we have, for vegetative organs, we have roots, shoots or stems, and leaves, roots, shoots, and leaves. And then for reproductive organs, we have flowers and fruits. And so really there are just 5, which is kind of nice. Not a huge amount to know. When we do all of these, we want to know primary function and secondary function. That's important. Okay, so let me just do a screen sketch of this, and we can think, okay, what about roots? What's the primary function? And then we'll talk about secondary functions. Anchor. Oops. Anchor to substrate. And you know, usually the substrate is the earth, right? It's dirt, but it could be a rock, or maybe it's, who knows what it's rooted on, bedrock. Okay. What else? That's a primary function of roots. The other very important primary function of roots is absorption. What do plants absorb? Water and nutrients. So the roots are very well adapted to absorbing water and nutrients directly from the earth, from the soil. Or if it's a water plant, they can absorb them straight from the water, right? And then it also anchors the plant and supports it. Okay, so we have that. Now, if we start thinking about roots and how they grow, if you think about a big oak tree, let's say, one thing you might think about is, you know, how far do the roots go under the ground? Let's say the canopy, you know, goes out here. Throw a few leaves on my oak tree. You get the idea. How far do you suppose the roots go? And how deep? You might be surprised to know that in most trees, the roots don't go very deep into the ground. Usually about the first 3 feet of the earth is where you'll find the roots. There are obviously exceptions. Some plants are strongly tap-rooted, and they might have a root that goes much deeper. How about how far do they creep out? Well, it turns out they go further than the canopy, usually 1.5 times the canopy. In this direction. So you can bet if there's a big oak tree in your yard, then those roots are going all up underneath your house. Okay? Makes sense, I hope. So there are 2 basic types of root systems. We could think of it that way. And one is called a fibrous root system. And so what you're looking at here is a coleus plant, and in the coleus, or in a fibrous root system, there are many roots and they're all roughly the same. There's not big ones and little ones per se. They're all roughly equal one to the next. Okay. Compare that to a taproot system. You're all familiar with a taproot system because a carrot is a really good example. And so in a taproot system, you have this one central root, and then branching off of this central root are lots of smaller roots, which we often call rootlets. Okay. And they don't always have to have this exact form. For example, in the case of a carrot, depending on the type of soil that you grow it in, and clearly they grew this just to show these rootlets here. But one of the things is if the root hits a rock or something like that, it will often branch. And so you could have branched carrot roots, but for the most part, it has a taproot. Okay. Things like carrots and parsnips, beets. Okay. Now, what we want to do at this point is we want to look at roots. We want to look at them in cross section and then also in longitudinal section. So we want to think about now, we have to think about tissues. We're looking at this thing anatomically, right? And so let's think about the different tissue types we've already talked about, right? We talked about parenchyma, xylem, and phloem, and we're going to see actually all of these different tissue types in here. So we start at the outside, and like just imagine this is a carrot. It looks kind of like a carrot cutting cross section. Epidermis. All plants are covered, all the surfaces of all plant parts are covered with an epidermis or a skin. What do these cells look like? They look like those very thin-walled cells, and in fact, this is all parenchyma. So think about when you eat a carrot, you know, this sweet part of the carrot is this part here, and it's parenchyma storing water and sugar, and some starch, okay? Now we have several layers in here that are difficult to see. I'm gonna draw them in. I'm gonna draw a layer here. It's a single layer of cells. And I'm gonna draw, let me do a different color. Let's do this color, what the heck. I'm gonna draw this, okay? And then I just wanna point out, we have this in the middle, right? And then we also have these little bunches of cells on the corners of this sort of star like this. There are a bunch of cells in there, okay? So that's actually kind of nice. We can see all these different tissues if we draw them in. So here's what they are. This one is called the endodermis. And this one is called the pericycle. Strange names, I know, pericycle is a strange name. The pericycle, it turns out, is a cambium. What does that tell you? A cambium is a meristem that increases girth. This is what lays down new vascular cambium, okay, and we're going to get to that. The endodermis is like the security system for stuff that gets absorbed by the plant and is trying to get into the vascular tissue in here. And so we're going to talk about how the endodermis kind of scrutinizes, and we'll talk a little more about that in a second. It scrutinizes what can get into the vascular tissue, because think about it, if it gets in the vascular tissue, then it has access to the entire plant, right? If it gets in the phloem, it can go up or down. So what's this? This is phloem. And this is xylem. And keeping with our you know, what we talked about before, phloem is to the outside, xylem is to the inside, okay? So there's all those different layers. And the one that we need to talk a little bit more about ends up being the endodermis, okay? So let's just take a second to do that. I hope y'all are enjoying this lecture. I know these are difficult times for everyone, and I hope you're all well. I'm a little bit tired. I had a big day between the flounder catching and the paddle boarding with the dolphins. The one good thing about teaching online is I can be wherever I want. So that's been kind of a blessing. But I do miss my students. I miss seeing your pretty faces all the time like I used to get to do. Okay, so endodermis. It's really made up of a series of cells. And let's see. So let me just draw out the xylem and the phloem, just so you know where we're at. There's the xylem, and then we have this phloem. I'm not going to put the pericycle in there. There's this. And then we have this, it's a single layer of cells, which is very strange. And I'm going to draw these. I'm going to take a second to kind of draw these. I'm getting better with this drawing stuff on this board here, I have to say. Getting pretty coordinated. Maybe I spoke too soon. Okay, so this is the endodermis. Okay. It's to the inside xylem and to the outside phloem. This is the endodermis. If you looked at one of these cells, you'd see it's three-dimensional. It's a box like this, right? It's very much of a box of a cell. And here's the crazy thing, y'all. The side walls, like this side wall and this side wall, and the top and the bottom are all covered with a waxy substance, which is called suberin. Okay, it's very thick. There's a side and the top and the bottom, okay? And so the only walls that aren't covered are the front face right here, and then the back face back here. And I know it's three-dimensional, it's a little hard to see, but if you can visualize it. What this means is that anything that's passing from the parenchyma out here through the endodermis and gaining access to the vascular tissue literally has to pass through the front of the cell and out the back, okay? And so what then happens is these cells actually are able to scrutinize, and so here comes stuff and it's going through like this, okay? This is the bottom, and this is the top, and there's one side and one side. So what this cell can do is it can scrutinize what gets into the vascular tissue. So it might not allow certain things to go in. It's pretty good at filtering out things like bacteria and large particulate matter. And so because of that, you can safely drink fluid out of a plant, and it's relatively pure. You wouldn't expect it to have any sort of bacterial contamination. Okay, so you can think of the endodermis as the security system of the root and keeping stuff from getting into the plant. Okay. Okay. Let's see what now. Now we can talk about secondary. Oh no, actually, I'm sorry. We have to think about a longitudinal section of a root now. There's a lot of morphology here, but since we're not really getting to have lab with microscopes, this is good. So we have these different, now think about your finger. You know, before we were looking at it, if you cut your finger crossways, Now think about it, if you cut your finger long ways, like this is a longitudinal section, there's the tip of the root and it's in the long section. And in this case, we have these different zones. We have at the tip of the root cap, and we have the zone of cell division. Well, you know what this is, mitosis, right? This is where more cells are being made. This is basically the RAM, the root apical meristem that we talked about before, right, in this region. And then these are cells that are the products of this mitotic division, and this is where they get longer. So the cells literally start to elongate. And then even further up is the zone of cell differentiation. And so this is where you get things like xylem and phloem. You get differentiation means that these cells begin to differentiate into the kind of cell they're going to be when they grow up, like a xylem cell or a phloem cell or a parenchyma cell. Okay, so anytime you make a longitudinal section of a root, you can always see these different zones that are present. You can't necessarily tell where one stops and one starts, I will tell you that, but it would be good to know them in order. Maybe I'll say name the 4 zones in order from the root cap up, you know, something like that. Now the root cap is especially interesting. It is responsible for the gravitropic response of roots. And what we mean by that is there are special cells in this root cap that can detect gravity, and it's remarkably similar to the way that to the cells that are in your inner ear that can also detect gravity. If you looked at these cells, they're boxy cells like this, and they have little hairs at the bottom, and floating around inside the cell are starch grains. This is starch. And so the way the root knows what's up and what's down is if the starch grains are stimulating the hairs, then it knows that it's upside down, okay? So roots will always grow downward. It doesn't matter how they come out of a seed, they always grow down because they have this gravitropic response, okay? The zone of cell differentiation is also important because it's the root hair zone. And a root hair is an extension of the epidermis of the root. So it's really cool to see under the microscope. A lot of times the nucleus will mosey on out into this hair, the nucleus of the epidermis cell. So a root hair is literally one cell. It's an epidermal cell with this extension on it. What's the point? Well, increased surface area, right? And we see this in we see this over and over in science. If you want to increase surface area, just project something outward. And so we'll say a couple of words or a few words about roots and their ability to absorb. They've done studies, a lot of the studies are done with rye plants. Rye as in the grass. And so let's say you got this grass plant. I don't know why it's not green, it should be. That's okay. And you got all these roots. And off all the roots, you got all the root hairs, right? You get the idea. Well, how many roots can one ryegrass plant produce? 40 billion roots. Holy moly. How many root hairs? 60 trillion. And then you start thinking, okay, well, what's the surface area of all that? If you iron, it's all three-dimensional, right? Everything's, look at your finger. It's three-dimensional. It's like your finger. If you ironed it all out, it would have the surface area of a football field. And so that gives you the idea of the incredible capabilities of absorption for roots. Okay. All right, team. doing good. We're going to keep going. Okay. Now we think about secondary functions. Well, there are lots of secondary functions of roots. One of the really important ones is prop root, but there are other. For example, a cornfield gets blown over in a big thunderstorm or something like that, and you think, oh, poor farmer, all his corn is laying down, but within a matter of days, it will literally prop itself back up, right? And that's because of these roots that are gravitropic and detecting that it's sideways, and then it pushes itself back up. Okay. Storage roots are obviously very important. It is important to note that yams and sweet potatoes are, in fact, good examples of storage roots, but that what about an Irish potato? Is that a root? No. Irish potatoes are actually stems, not roots, okay? But yams, in fact, are roots. What are some other ones? Beets? Carrots, lots of other ones, right? Turnips, these are all storage roots. Think of other stuff. Roots are these are roots which we, or in this case, the branches. This is a strangler fig that you see here, and this is a really dramatic example of aerial roots. They call it a strangler fig because typically what happens is this tree will start to grow, a seed will germinate, usually they're spread by birds, and it might germinate in the crown of a palm tree. They're tropical trees, so imagine a big palm, and this little tree might start to grow right here in the canopy, and it will grow into a strangler fig, and then as its branches come down, it will literally strangle this tree like this. And so, you know, maybe somewhere right in here is where the palm tree was, and now there'll be a hole in here where the palm trees died and filled this space. It's really dramatic. Okay. Aerial roots are sometimes called adventitious roots. You can also see them on poison ivy, which is cool. Let's look at that real quick. This is interesting. Oops. Yeah, here we go. This is a great image of aerial roots or adventitious roots. Oops. Sorry. Let's look at this one. See all these masses of roots? This is not a, it's not a parasite. It's not taking anything from the tree. It's just using these roots to grow up the tree. Okay. Good to know. And then nematophores is the last one that we'll look at. Sorry, I'm just checking the time. Okay, we're good. Nematophores, you see these typically in swamps, areas where plants are growing in water, and nemato, air, right? And what these do, it's believed, is provide gas exchange for the roots, because the roots are growing in water, there's very little oxygen. And so they need to have a way of getting oxygen. So O2 is going in and CO2 is coming out. And so it's a way to do gas exchange, because the roots are living cells and they have to be, you know, they have to have a way to get oxygen, and they have to have a way to get rid of CO2. So we see these in swampy areas. Mangrove is a really good example, and that's what you're looking at here. This is the kind of a plant, okay? Good. And I think that's probably the last slide for roots. That brings us to stems. And we might as well go a little bit further, see if we can get through stems. What do stems do? Well, pretty straightforward. There are secondary functions of stems, but primary function of stems is support the leaves, which are the photosynthetic part of the plant, and transport, right? They transport water up to the leaves. They transport sugar up and down. And they also support the photosynthetic organ or the leaf. And now we have to take a little time out because we need to do some, kind of a little sidebar, a little diversion for just a second. And this will actually be the last slide that we'll look at because we're going to do a little chalk and talk here for a second. There are ways that you can kind of categorize or classify plants, okay? And we're going to look at some different ways to group plants, okay? And one of the ways to group them is whether or not they are woody or herbaceous. These are kind of two general ways. You can take any vascular plant, you can say it's either woody or it's herbaceous. That's one way to lump them together. Woody plants, by definition, have some kind of persistent above ground vegetation. Trees and shrubs, right? There's always something above the ground that's alive. Think about trees. I mean, in the winter, some of the trees here lose their leaves, but there's still above ground portion that's alive. Same with shrubs. Herbaceous, no persistent ... right? Above ground vegetation. Okay. Can you think of an example of an herbaceous plant? Sure, of course. Lots of examples. We'll get to that. Another way to group plants is by their life cycle. Are they an annual? Or are they biennial? Or are they perennial? I think there's two N's in that, but I'm going to leave it like that. I'll stick another N in there. There we go. Perennial. Oh, gosh. I'm sorry. I don't think there is. Okay. What does this mean? How long? It has to do with how long it takes for the plant to grow and complete its life cycle. Annual plants live 1 year. They start from a seed, they grow vegetatively, they flower, they fruit, they make a crop of seeds, and they die, all within 1 year of time. Okay? Biennials, bi means two. These take 2 years to complete their life cycle. And then in a perennial, I think there's two R's. It's either two R's or two N's. We'll look it up in a second. These have indeterminate lifespans. They just grow year after year, right? And they just keep coming back, okay? So, you know, here's the thing. Now we can put these two words together and we can say, oh, it's a woody perennial, or it's an herbaceous annual, or an herbaceous biennial, okay? A good example of an annual corn. Doesn't matter where you plant it in the world, it grows, it flowers, it fruits, all within a year. Good example of a biennial, carrot. Carrots we grow as an annual crop, but if you leave them in the ground, the spring, there's the carrot, the spring of the second year, it will send up a flower and then it will die. And that's the end of the carrot, okay? So they live 2 years. Perennial, how about an oak tree, you know, year after year after year. So let me ask you this. Can you have an herbaceous perennial? Does that make any sense? It does. It does make sense. Herbaceous perennial. Herbaceous, no persistent above ground vegetation, but perennial, meaning it lives an indeterminate lifespan. Example of an herbaceous perennial. Dandelion. Think about it. The dandelion comes back every year in your yard in the same spot. It survives the winter with its perennial taproot, right? So you can have an herbaceous perennial. Can you have a woody annual? That doesn't really make any sense, right? Because if it's woody, it has persistent above ground vegetation year after year, but if it's an annual, it only lives a year, so that doesn't really make any sense, okay? There's a third grouping, monocot versus dicot. The word monocot and dicot come from these little structures called the cotyledons. And if you see a dicot, think about when you plant beans in the ground, they come up out of the ground, they have 2 little cotyledons. These are the seed leaves, okay? Monocot means one, mono has just one. Okay, so beans are dicots. Corn is a monocot. You could count the cotyledons and that will tell you if it's a monocot or a dicot. But that's not always going to be practical. I mean, who's going to just happen to be there when the little seedling is germinating, right? There are other ways. Look at the leaf structure. Specifically the veins, and in a monocot, the veins are almost always parallel to one another in the leaf like this. They almost always run in a parallel fashion. Okay, in a dicot, the leaves, there's a bean leaf, and they have what we call a reticulate veination, meaning the veins branch. There's often a central vein, and then those veins branch and branch further still. We call that reticulate. Think like a reticulum is like a spider web. Okay. You can almost always, you can be walking around outside, you know, look at a leaf and tell if it's a monocot or a dicot. Almost always, 99% of the time, it will be accurate just by looking at the way the veins are in the leaf. It's a really good way to tell. One other way, flower parts. Like, for example, petals. In monocots, they're almost always in 3s, and in dicots, they're almost always in 4s or 5s. This one doesn't always work. But if you combine it with the leaf vein thing, you can 99.5% of the time tell a monocot from a dicot. What I mean is maybe it has 3 petals or 6 petals or 9 petals. Or maybe it has, if it did, it would be a monocot. Or maybe it has 4 petals or 8 petals or 5 petals or 10 petals or some multiple of these. Okay. So there's all that. And let's see where we're at with the time. Yeah, I think we're kind of to the end of what I wanted to cover today in this PowerPoint. And we'll come back to the monocot versus dicot stem slide next time and see how There is, in fact, a fourth way to tell a monocot from a dicot. If you're walking through the woods and you happen to have your handy dandy microscope, you can make a cross section of a stem and look at the vascular bundles, is what we're looking at here. So we'll come back to this slide next time. Okay, and it's been fun teaching you all so far. I hope you're learning and enjoying. Feel free to watch the video over and over. I'm sure you're all going to do that. Otherwise, I will see you next time. Take care. Sayonara. Adios. Bye-bye.